So adaptive immunity, in contrast to innate immunity, which was what we left off with. Innate immunity being the physical barriers, the physical processes that we've had since birth, that we'll always have, that are broad spectrum combat or responses to invading pathogens. Adaptive immunity is different, though. The magnitude of the response can change. The specificity and the targeting of the response can change. It can adapt. And adaptive immunity deals with that concept, that you can become more resistant to something if you've been exposed to it. So we will follow suit with a few of the animated videos from Wiley Plus, and then elaborate on those concepts and topics as we go along. There's a really good section on adaptive immunity that you have access to, that you have access to through Wiley Plus still, if you ever log into that anymore.
Our skin and mucous membranes normally provide a barrier against invasion of foreign agents. If damage to our skin occurs, local defense mechanisms mount the initial attack on invaders. If deeper penetration of pathogens occurs, the adaptive immune system relies on lymphocytes to provide a two-pronged attack on invaders and infected body cells. One response, cell-mediated immunity, occurs when T lymphocytes directly attack and destroy diseased or foreign cells. The other process, antibody-mediated immunity, does not involve direct lymphocyte destruction of invaders. Instead, B lymphocytes release antibodies into the blood and interstitial fluid to neutralize pathogens. T lymphocytes mature in the thymus to become either cytotoxic T cells, which directly attacks diseased or foreign cells, or helper T cells. Helper T cells aid the immune responses of both cell-mediated and antibody-mediated immunity. B lymphocytes mature in the bone marrow to become plasma cells, which produce antibodies. During the maturation process, these lymphocytes develop the ability to carry out adaptive immune responses through the insertion of distinctive proteins, called antigen receptors, into their plasma membranes. B lymphocytes, when activated by the secretions of helper T cells, divide by a process called clonal selection into antibody-producing plasma cells and B memory cells. Cytotoxic T cells are similarly able to clone themselves into active cells and memory cells. Helper T cells have a central role in the immune response by releasing stimulatory molecules known as interleukins. These interleukins greatly increase the cloning of plasma cells to stimulate a high production of specific antibodies and cytotoxic T cells to attack invaded cells. The number of memory cells also increase substantially. Most active cells eventually die after the immune response has been completed. Memory cells remain inactive in case the same pathogen enters the body at a later time. This enables them to rapidly respond to the next invasion.
Ooh. That was 2 minutes and 35 seconds, but it felt like 10, right? We hit the ground running. There's a lot of information in that video. We are going to elaborate and figure out what's important and come back to those topics over the next few slides. So we saw different types of adaptive immunity. We don't know what they really mean yet, but you saw examples on the slide. We'll elaborate. A little bit of review as well. Remember, T cells become immunocompetent in the thymus. We talked about that last class. And B cells originate from the red marrow of long bones. So there are a lot of different variations of those cells. There were a smattering of different names on purple balls, on some that had projections coming out of them. There's a lot of different cells or variations of cells that we'll get into. It's not important that you remember them or know them right now. But let's figure out what's important about this video that we want to reiterate and bring back up. on the concept of adaptive immunity. So this simply refers to the body's ability to target a specific pathogen or to target a specific microbe. And that idea of specificity, a specific antigen or a specific particle, is one of the hallmark characteristics of adaptive immunity. It is specific. Innate immunity is not specific. It provides an equal barrier to all things equally. Equal barrier to all things equally. Adaptive immunity is specific for a particular antigen. And it remembers that antigen. It responds in some manner on the initial invasion, and then on subsequent invasions, it can remember how that first experience, that first encounter went, and adapt more quickly or respond more quickly. So it has a memory allowing a more vigorous response to previously encountered antigens. Now I want to make clear, we've used the word pathogen before, we're using antigen now. Antigen is simply the part of a pathogen that triggers the response. It could be the whole cell. It could be a bacterial cell. It could be a protein. It could be a toxin that's released. But it's the thing that triggers the response. An antigen is actually a mashing up of two words. It comes from antibody generator, antigen. That's where the name comes from. It's the specific part of a pathogen that triggers the response, that generates antibodies, hence the name antigen. So you can think of antigen and pathogen almost interchangeably, but know that specific difference. Antigen is the thing that the immune system is responding to. It's more specific. So how does the body respond to invading pathogens that display these antigens? Here, B and T cells are the stars. They're the stars of the adaptive immune system. When they are fully differentiated, when they are immunocompetent, they display receptors on the cell surface. And these are sensors that are waiting to pick up signals in the body that there's an invading pathogen. They're looking to bind to the specific parts of the invaders to trigger an immune response. And the schematic on the right shows a lot of detail, and it's up there for reference so that when I post these notes, you can look through it in a lot of detail. But just notice right now the source and the types of immune cells involved in the response. So red bone marrow on the right, the thymus on the left, giving rise to B cells and T cells in their variety of flavors. What are those flavors? B cells and T cells are the stars of the show, but they are the umbrella term. They're the classes of immune cells. Specifically, the types of T cells that we want to see in response to an antigen are, number one, cytotoxic T cells. Cytotoxic T cells. Sounds like they might have a role in carrying out an immune response. They might be toxic to the cells, cytotoxic T cells. And helper T cells. And I've asterisked helper T cells to denote their importance. They are typically the cells that turn on the other cells. And you saw that in the video with the secretion of interleukins, which is just the signal to turn on other cells. Helper T cells are integral in activating cytotoxic T cells, pointing them in the right direction, saying, attack that one. Helper T cells delegate. Think about it that way. Helper T cells don't only activate cytotoxic T cells, they also activate B cells or plasma cells. B cells, when they're immunocompetent, are called plasma cells. They are also directed and turned on by signals from helper T cells. So these are the three main players in the adaptive immune response. Helper T cells activate the other two, cytotoxic and plasma cells, and those have specific roles that we'll come back to later. Now, what I also want to mention, well, first of all, you can see helper cells in the middle of this figure on the right-hand side with this really nice descriptor showing help. To the left and the right, both the B cells and the T cells are being helped by these helper T cells. They're being activated or turned on. Those signals are being sent to delegate and dictate what the roles are. But I want to point out that both types of cells, all types of cells, make memory cells. When they divide, two things are produced. Let's take a cytotoxic T cell, for example. When it divides, it makes another cytotoxic T cell, and it makes a memory cell. And all of these cells do the same thing. A helper T cell will make another helper T cell, and a memory cell. Plasma cell will make another plasma cell, and a memory cell. And that's a really important feature in the memory aspect of innate immunity, being able to recognize and respond to an antigen 10 years down the road. It's because of the memory cells that are produced that we have that long-lived ability to ramp up our immune response, that we have that memory. So all types create memory cells. Now, if we think about the helper T cells as pointing everyone else in the proper directions, what are we left with? We have cytotoxic T cells and we have plasma cells. There are two workers. And conveniently, there are two types of adaptive immunity. And you would be right to think that one type of cell is primarily involved in one type of adaptive immunity. So you heard reference to cell-mediated immunity. This is one branch of adaptive immunity. And this is mainly carried out by those cytotoxic T cells. Cytotoxic T cells carry out cell-mediated immunity. What does it mean, cell-mediated immunity? This is the kind of response that is required when a cell in the body has been infected. If the immune system has to get into a cell or break up a cell, destroy cells, cell-mediated immunity is required. And cytotoxic T cells are the things that will do that job. On the other hand, the second branch of the adaptive immune system is antibody-mediated immunity. Antibody-mediated immunity. This is the branch of the adaptive immune system that ramps up in response to antigens that are outside of the cells. If they're in the blood, in the plasma, if they're in the interstitial fluid, antibodies are released to target those things that are floating around freely. And antibodies are primarily released from B cells, or plasma cells more specifically. So two arms of adaptive immunity, two main types of lymphocytes, one type per arm. Make sense? An important thing to bring up that's not on this slide is that, let's say you have a bacteria that's invading your body for the first time. Any pathogen that invades for the first time, or any time, is likely to induce both arms. Okay? First exposure, there's probably a lot of the toxin or the pathogen floating around in the blood. Some cells might start to be infected, but both arms of the adaptive immune system will be activated. It's not just one or the other, both are activated similarly. It depends on how much of the antigen is circulating free and how much is within the cells, but by and large, it's not one or the other. Both happen. Okay? Now, the process that we described on the last slide of B cells proliferating, or plasma cells proliferating into plasma and memory cells, or any of the other lymphocytes that divide into a memory cell and another active cell, is called clonal selection. It's a special kind of division where the type of cell that you started with is cloned and a memory cell is produced. So clonal selection is ramped up with any invader. Between sicknesses, between invasions, between immune responses, there are generally there's a smaller number of lymphocytes present. And a smaller number of lymphocytes means a small response at first. We don't want a small response, especially if it's important to get rid of a toxin or get rid of a bacteria or get rid of some other foreign invader. We want a large response, and we achieve that through the process of clonal selection. We make a large pool of lymphocytes. They divide and divide. An equally large number of memory cells and active cells. But having that large number of lymphocytes allows for a large immune response. And it's through the process of clonal selection that we get a large number of active cells and a large number of memory cells. This allows us to initiate a strong immune response and react accordingly. The reaction so far, we're not sure what it is, but we'll see that coming up. So, this describes a little bit of a lag period. For a few days after you get a cold or some invading pathogen, we are busy making this large pool. And then after a few days, the pool starts to mount an appropriate immune response. That's why it seems to take a while to respond to a cold or a flu or some other invading pathogen. These memory cells are the ones that are responsible for the rapid response later. They can live multiple decades, 20, 30 years. So you hold on to that ability to ramp up your immune response for multiple decades after an initial infection. They don't live forever, which is why in some cases you need a booster. Booster of what? We don't know. We'll talk about that later on too. But to induce more memory cells to proliferate through this process, clonal selection. So I think we have a good grasp on at least the branches of the adaptive immune system, the types of cells that are involved. We know the general roles. We don't know the mechanisms yet, but we know that cytotoxic T cells are assigned to cell-mediated immunity. Plasma cells are assigned to antibody-mediated immunity. We'll look at both of those in a bit more depth after we talk about the responses in general.
Pathogens such as bacterial cells or even cellular components that are recognized as foreign provoke immune responses and are called antigens. Small parts of a large antigen molecule, called epitopes, act as the triggers for immune responses. Each epitope on an antigen can activate a specific T cell, or induce production of a specific antibody from a B cell to target different antigens on a single pathogen. An amazing feature of the antigen receptors on both B and T cells is its ability to recognize and bind to at least 1 billion different epitopes. The T and B cells are able to differentiate between healthy and diseased body cells, or foreign cells, because all body cells, except for erythrocytes, produce a membrane marker known as the Major Histocompatibility Complex 1, or MHC1. When a body cell becomes infected by a pathogen, the pathogen's antigen is added to the MHC1 marker, creating a signal that targets the infected cell for destruction by immune cells. If the pathogen has not invaded the body cell, defensive cells, including B lymphocytes, phagocytes, and dendritic cells, known as antigen-presenting cells, or APCs, consume and digest the invader. Fragments of the pathogenic antigen are attached to a second version of the MHC identifier, MHC2, which is inserted in the APC's cell membrane. These cells expose the antigen in a way that activates helper T cells, which in turn promote a higher level of adaptive immune response.
Just the major histocompatibility complex 1, right? You knew that. Okay, so we're getting some information on how these cells recognize invaders. So far, we've just understood, oh, they do. But how do they recognize invaders? And how do they differentiate invaders from self, from my cells? How do I keep my immune system from attacking my cells? And it turns out that there are many different features that could initiate an immune response. One pathogen can have a multitude of different triggers, of different antigens. And the specific sequences are called epitopes, these small little amino acid or protein bumps or parts of a protein that trigger and bind to antibodies or bind to T cells. There can be many of these on a given antigen. Now, when an invader comes into the body and initiates an immune response, it's said to demonstrate immunogenicity. And I bring this up, it's a matter of semantics, really. But an antigen that prompts an immune response is demonstrating immunogenicity. And after the immune response has been initiated, when the antigen is a target of the immune response, they are demonstrating reactivity. So in one sense, in the first sense, they are the effector, they are causing an action. And in the second sense, they are the subject of the action. They are initiating an immune response, and then they are the subject of, the target of that immune response. This is one distinction I don't think the book does a great job of trying to differentiate, but we're not going to go into too much depth about it. We just need to know that there is a way to identify an antigen, there is a way to respond, and there's a way to protect the rest of the cells in the body from that response. So, the word antigen, we understand as the specific part of a pathogen that triggers an immune response. It turns out that specific part can have specific parts. That's what the epitopes are. The small features of an antigen to which antibodies bind, or to which T cells bind. It doesn't keep going, it stops there. There's only 2 layers, which is good. But we're getting a sense of what the mechanics are behind this response. So any invading pathogen will have a number of antigens on it. Every antigen has a number of smaller bumps or epitopes. All of these have the ability to trigger or bind cytotoxic T cells or plasma cells. More specifically, antibodies, but we'll get into that. So we need to be able to register, to recognize, and to bind these epitopes. The division, the distinction here holds up. Cell-mediated immunity carried out by cytotoxic T cells is done when cells have been infected. Antibody-mediated immunity carried out by plasma cells when there are antigens circulating freely in the plasma or in the blood. And so the antigens that are to be recognized need to be in those spaces. It's easy to understand how an antigen floating freely in the blood can be recognized. It's right there. I can grab it. It's easy to understand how the immune response is triggered in response or on the antibody-mediated side. It's more difficult to understand how an infected cell is recognized. Aren't cells that are infected still my body cells? Where's the switch? Normally, my immune system isn't going to attack that cell, but once it gets infected, it will be attacked. How is that possible? The answer is through MHC1. That's what we'll refer to it as, the Major Histocompatibility Complex 1. Everyone's is slightly different. Even identical twins have different Major Histocompatibility Complex 1s. And this is a protein that's in every cell membrane in the body except for red blood cells. So it labels every single cell in your body and says, this is mine, except for red blood cells. Which is actually a really important feature and explains why transfusions are successful. Blood donation, they're not labeled as being foreign or someone else's. They have another kind of label that we're not going to talk about, but they're not targeted by the immune system, which is great. So normally, MHC1 says, don't attack, I am a friend. I'm part of the body, leave me alone. But MHC1 can be modified when that cell is infected. And that modification will bring in the cavalry. The immune system will target it appropriately when the cell is infected. This is a big reason why transplanted tissue can be rejected. That tissue will have a different MHC1. The immune system will think it's infected and try to attack it. So how? On the cell-mediated side, cell has been infected, T cells are invoked. How are T cells signaled to target the infected cells? The thing that has done the infecting, we try to break down and degrade. We can digest it still. There are still aspects of the innate immune system that are digesting those invaders. And parts of those invaders that have been digested can be mounted for the immune system to see. Like a severed head on a steak. It's a warning signal. It's a pretty severe example, but I've been trying to catch up on Game of Thrones before the new season comes out. Like a severed head on a steak, part of the invading pathogen will be mounted for the immune system to recognize, to target, identifying that cell as having been infected. So antigens are continually broken down, and parts of that broken down antigen are mounted on the major histocompatibility complex. The MHC binds with part of the antigen, forms a new complex, and this is presented to the immune system. Let's take a look at how that works in the cell. This can happen in two different ways. If we have an invader, a microbe, that is traveling around outside of the body, the antibody-mediated immune response is active, it's ongoing, but we don't want to rely only on that branch of the innate immune, or the adaptive immune system. We want to also invoke cell-mediated immunity, and so to do that, we can take any of these circulating antigens, exogenous antigens, which simply means outside of the cell, and eat them. We have specialized cells whose job it is to ingest and digest these circulating microbes. And these circulating cells are called antigen-presenting cells. When we looked a couple lectures back at the lymph nodes, I made note that there are B and T cells, and there's macrophages and dendritic cells. I said we'd come back to that later. The dendritic cells are important. We'll see that later. Dendritic cells are an example of an APC. They will track down microbes, ingest them, break them apart. They are an antigen-presenting cell. And this slide has a lot of detail on it, but we've seen the majority of it already. This is a lot like the process of phagocytosis. A microbe is brought in, it's put into a vesicle, it's digested or degraded. Instead of just being expelled, we take those breakdown products and we do something special with them in APCs. That thing that we do is we want to signal the rest of the body that antigens are present, mount an immune response. So we make a small variation of MHC1, here called MHC2, very similar protein, whose sole purpose it is to bind the breakdown products of that microbe. And once that's bound, that new complex is pointed outwards, head on a stake, so that immune cells in the body can recognize it and say, oh, this antibody is present. This antibody needs to be targeted. This antigen is present. This antigen needs to be targeted. and I need to mount an according immune response. So this is what we mean by antibody presentation. Antigen presentation. I'm going to have to watch that so it's not so confusing. Antigen presentation. They are presented externally to elicit an immune response from other B cells and T cells in the vicinity. So cell-mediated immunity can be activated in this sense. We can devour some microbes in the circulation and present them for action by the immune system. Or, the counterpoint to this, these were exogenous antigens. If a cell has been infected, we need a similar method of response. This is a slightly different scenario. Here we have one infected cell. The antigen has made its way into a normal body cell, not an antigen-presenting cell. The biggest difference between this and the last example is just that. This is a normal body cell. It doesn't have the capacity to make a new MHC and present the complex to the immune system. This is a normal body cell which will co-opt, it will use the mechanism it has in place, the normal MHC molecule which identifies it as body, self. A normal cell is modified as a flag to say, I've been infected, which is a signal of self-sacrifice, really. As soon as this signal is registered by the immune system, this cell is targeted for destruction. And that's the topic of the next section that we're going to look at really quickly. So the methods of destruction, the methods of breakdown of these antigens is what is coming up in our exploration of the two arms of the adaptive immune system. So in both cases, whenever an antigen gets into a cell, it's presented outside of the cell for the immune system to react accordingly. Otherwise, as we'll see in the video that I want to show you coming up, otherwise, viruses can proliferate, bacteria can proliferate inside the cell, cells can grow and divide, and in unregulated, massive, extreme circumstances can lead to things like cancer, where we have massive proliferation of infected cells or altered cells. So let's take a look at each arm of the adaptive immune system and how it might work. How antibodies work to break these things down, how cytotoxic T cells work to blow apart this cell in response to a signal like this one.
When a cytotoxic T cell is activated by the actions of APC and helper T cells, it recognizes the MHC1 complexes on the infected body cell's surface. Cytotoxic T cells then release two molecules, perforin and granzyme, in order to destroy the infected body cells and the pathogens that infect them. Perforin creates pores in the target cell membrane. The pores allow extracellular fluid to enter the cell, causing it to swell and potentially rupture the cell membrane. Granzyme then enters through the pores and causes the degradation of its proteins to ensure cell death. Cloned B cells do not attack individual cells. Instead, after they become plasma cells, they greatly increase their production of antibodies. These antibodies are released into the plasma and extracellular fluid, where they will match and bind to the target antigen. Antibodies are Y-shaped structures with unique antigen-binding sites on the tips of two of the arms. This allows an antibody to perform different functions depending on the type of binding site it possesses. They may essentially prevent invaders from interacting with normal body cells, or mark foreign cells for destruction, and activate our defensive cells into specific action.
So, we're getting insight into the mechanism of these two arms. Now,